Please welcome onto the stage, Andrew Hanson. At the end of this talk, you may be convinced that the four colours shown here are identical. Colour is that crazy. So let's press on. Does anyone remember the dress? Big social media thing about 3 years ago. Somebody shared an image of their wedding dress and the world went wild, arguing about its colours. We'll go into that. I bought the dress. My daughter was too young to fit into it, but there she is standing next to it. And I took a photograph. I illuminated it brightly from one side and dimly from the other. And in one photograph alone, we can pull the bits out and we can see that indeed, under some conditions, it looked white and gold, and under other conditions, it looked blue and black. A conference was held to discuss all the various implications of this. The Colour Group of Great Britain, all of their committee were approached by a number of media outlets. I was approached by the Telegraph. I had a column written in there. My mother was delighted. My father was disgusted. He said, "You should have been in The Guardian." Before I move on, I'd like to tell you who I work for. The National Physical Laboratory, or NPL as it's known in short, is the National Measurement Institute for the United Kingdom. We have about 1,000 people all measuring stuff on your behalf. When it was set up about 100 years ago by the Royal Society, it was actually, well, they call it the National Physical Laboratory. I think they should have called it the National Measurement Laboratory. We do physical measurement there. And for 20 years of my life, I was engaged measuring colour and light. And that's the basis of what I'm going to talk to you about today. But the building itself, well, it keeps everything running for us. You may not realise it, but we rely on measurement. Every aspect of 21st century living, and before, relies on knowing how much you've got of things. Nuts need to fit bolts. You need to know that the ingredients, the active ingredients in drugs are the right amount, and so they're measured really accurately. And most importantly, when you buy chocolate, you need to know you've got the right amount. So weights and measures and all sorts of other measurements are really, really important to make the world work. And that's what we're there for, doing it for the UK. Set up about 100 years ago in the corner of Bushy Park. And we moved about 10 years ago into a nice, big, new, glamorous, white building with 388 separate laboratory spaces, some of them very large indeed, with very odd things going on. All measurement, though. And since the 1910s, we've been involved in measuring colour and light. Electricity has only been brought into our houses for about the last 100 years. And it was brought in not to run television or iPads or heaters, but light. So measuring the efficacy of lighting systems has been in there for pretty much 100 years now. We use colour every day. And there's a photograph of the outside of this building. And in just one scene, we can see the use of colour in branding. And it's really important that those brands, once they've been sorted out, are shared appropriately with the right colours, that your web page has the right colours, that mugs and clothing and printed materials all look the same colour. So branding is really important, and we need to communicate the colour of these brands appropriately. Quality control. Imagine if your car got dented and needed a respray. You need to be confident that the colour of the respray fits with the original colour of your vehicle. And finally, legal and safety. There are lots of yellow lines and white lines out down this road just outside. And there are rules about how visible those those lines need to be. So people go out there and measure them all the time. You need that for visibility. We also use colour to manipulate emotion. So there's a lovely view graphic up there of all sorts of colours for brands. Maybe you recognise some of them. And according to the people who created this diagram, you can associate particular emotional responses with different colours. Gets you thinking, doesn't it? So there's a need to measure and specify colour. Let's quickly pick apart those needs. You create a brand or an identification system, which really needs colours to be communicated correctly beyond language to other parts of the world who might be creating things on your behalf. You also use it in monitoring processes such as creating paints, but also I bet if you go into a shop and you're faced with a huge amount of orange juice, if one of those containers contains something that looked slightly different in colour to the others, that's the one you wouldn't buy. So in monitoring processes, colour's used quite a lot. Now, if you're communicating colour, you need to talk about absolute values. You need to be able to recreate exactly a colour remotely, and that requires an absolute definition. But if you're monitoring changes in things, maybe you're just interested in difference values or things relative to the target colour. Now, these have their different requirements, but they'll still require some precision in colour measurement. Now, we are humans, and we have a visual response to light. Light has wave properties, and we can split light up according to the wave properties. So I've brought along a little demo here, which I hope will work. Can we dim the lights a bit, James? I've got a very nice, fancy light bulb here, and I'm going to dazzle the guys over there. There we go. Can you see that? Good stuff. So we have a mixture of white light coming on here, and this thing here acts a bit like a CD. Have you ever held a CD up to the light and seen all the bright colours coming out of it? It does the same sort of thing in a raindrop as well. And it splits the light up into its various components. So from this direction, we have the blue of a light-emitting diode shining through a phosphor that generates other parts of the spectrum through the process of fluorescence. And then up here, we've got the whole thing starting again, because that's the way this system works, unfortunately. So we've got the next lot of blue overlapping. But if we could just look at one spectrum remotely, just one spectrum, then you'd see it going from violet, blue, indigo, well, the colours of the rainbow. Let's see what I'll put up next. And our response to that spectrum varies. We don't see all colours evenly with the same amount of response. At the red end of the spectrum, the amount of energy in red light is not enough to excite the photochemistry of our eyes, in our retina. So we become blind to anything beyond a particular wavelength. We can't see infrared. Some animals can. At the other end, we can't see ultraviolet. Ultraviolet radiation has the potential to destroy the subtle chemistry and biology of our eyes, so we don't want it to come into our eyes. And the filter at the front of our eye is the ciliary lens. Something normally used for organizing light and focusing it also acts as a filter, so we cannot see ultraviolet radiation. Some animals can. And somewhere in between, at the peak of the green part of the spectrum, that's where our eyes are most sensitive to light. So as a physicist, I would say you need less watts of green light energy to excite a sensation of brightness in the eye than you need for the red and for the blue. You need lots more energy to excite the eye if it's red and blue. Let's look at two objects. I'm going to do the whole lights down again. I brought two objects along here. Can you kill the lights again? Thank you. Right, I'll see if I can get a slightly brighter spectrum. It's looking a bit dim to me. There we go. That's right. Oh, that's a bit better, isn't it? Let's do that one. Okay. Got two objects in my hand here. They're actually bits of gaffer tape. And if I go from the blue end of the spectrum, can you see they're both quite dark? But as I go for a little walk out here, cyan, green part, this top part that's a piece of green tape, looks really, really bright. As I move into the yellow, at some point, the two will appear equal lightness. Is that happening for you? And then as I go out into the red, the green part becomes dark and the red becomes violently light. Now, it's really interesting that people's responses to light are different. The cameras seeing this as well might see these slightly differently. So you'd all argue about which point the two look equally bright. That's quite normal. We are all individuals. Now, as a measurement scientist, well, that's great, but where are the numbers? And more importantly, where's my controller? Here it is. So we can measure things. And I said that light has a wave property. We talk about the wavelength of light. So I've written those along the bottom there. The numbers are actually 1,000 millionths of a meter, or nanometers as we call them. And we can only see quite a narrow band of wavelengths, from 380 in the violet to about 780 nanometers in the red. So we popped these two objects and we measured their spectral reflectance, how much light they were reflecting as a function of wavelength. And there they are. Can you see? Two lines. Now, the green line shows us that the green object was reflecting a lot of light at the green part of the spectrum, but not much at the other extremes. The other line, which I've made cyan so that you can all see them nice and clearly, is only reflecting light at the red end of the spectrum. Now, that property is something to do with this material. It's nothing to do with the light bulb, it's nothing to do with the way we see it. That's a property of the material. Let's have a look at the 3 stages of color production. You can have many, many different types of light source, all with different spectra. And they will shine onto different things that diffuse and scatter and reflect and absorb and transmit light differently as well. Again, with a spectral property. And then we resolve those spectra with our eyes. And if we need to, we train artificial intelligences or robots or cameras to see color the way that we do. So color is a 3-stage process. Something to do with the light source, something to do with something that changes the color of the light source by absorbing or whatever light, and then the way we see it as well, a 3-stage process. Now there's quite an odd thing that can happen. This observer is looking at 2 things and they appear the same, okay? They appear the same. And this observer sees them and they appear different. And the reason is that the eyes sense things differently. Okay, can you see that? And we call that observer metamerism. Now let's move to a different situation where you have one eye looking at 2 things and we change the color of the light source. Then we have a sample metamerism issue. And you might remember when cloth, you went into Marks and Spencer's or whatever and you compared 2 things and they would look identical in color under one type of lighting but different in color under a different type of lighting. I can see a lot of people nodding their heads. They'll only match under all the different types of light source if they actually reflect light identically. And it's quite unusual to get things to do that. So here's your typical O-level question, GCSE these days. If you have a blue object illuminated with a blue light, what color will it appear? Anybody venture an answer? Somebody said white, somebody said black, somebody said blue. I've seen the marking scheme. It says if you shine blue light onto a blue object, it will appear blue. That's supposed to be the easy question. Here's the harder one. If I have a red object and I illuminate it with blue light, what color will it appear? A unanimous black, fantastic. I guess you understand that red should absorb, or red objects should absorb the blue light, leaving nothing to reflect. And that indeed is what the marking scheme will give you 2 points for. But it's wrong. On 2 counts. What if the thing was shiny? It would reflect some of the light. There you go. And we call that specular reflectance. So you could argue that I'm just seeing the light itself, but you know, the thing is reflecting that light. So you could have a red object that kind of looks bluish because you're seeing the reflected light in it. a mirror reflection. What about this? It could actually look red because we have a thing called fluorescence. I'm sorry, I'm going to go back to this demo yet again because it's such a good demo, this one. Can we get the lights down again? This is the last time that we'll mess around with this. I've got here a yellowy object. It's a high visibility jacket. So I've got yellow light coming onto a yellow object and it looks yellow, okay? What if it was blue light falling onto a blue object? A yellow object. Oh my goodness, it still looks yellow. In fact, it doesn't really matter what I put this under, it will be a yellow of some type. It really doesn't matter. And down here, we have high energy blue light losing some of its energy in the chemicals inside here and re-radiating at lazier, lower energy yellow part of the spectrum. We call that fluorescence. It makes measuring an absolute pain because you put one type of light on and you get another type of light off, which seems just stupid and not in the marking scheme. What makes colour? Well, there's a great book called The Physics and Chemistry of Colour, The 15 Causes of Colour. It's by Kurt Nasu. Pretty much each chapter deals with a different reason for creating colour, and as a physicist I was quite upset by this book because it all seemed to be chemistry. A lot of it is to do with electrons and how they move, how they absorb, and how they radiate energy. But that's only half the book, there's the other half. And there's some physics creeping into the later chapters. Let's look at some of it in practice. In autumn, leaves go through a series of incredible colour changes, all to do with the chemicals inside. Now, normally you'll have chlorophyll in a leaf, very complicated molecule that wiggles at some frequencies and absorbs energy. It's using blue light, violet light, and red light, collecting energy at these wavelengths, but it's reflecting the green light. It's not using the green light at all. So we just see what the leaf doesn't need. As the leaf dies, we then see the carotenoids. Now, chlorophyll is actually quite a weak molecule. It'll fall apart quite readily. So they have carotenoids in there to mop up free radicals that would otherwise destroy the chlorophyll. So those chemicals are in there to keep the thing going. They're also the reasons why carrots are carrot-coloured. I guess there's a connection with the name. And finally, as the leaf dies and the chlorophyll stops working and the carotenoids stop working, we're left with anthocyanins. Anthocyanins are a set of dyes that change their colour with pH, from violet to pink. And it's no coincidence that the names violet and pink are colour names, but also flower names. So how do we see colour? I'm going to do a human experiment on you now. Can we dim the lights a little bit, James? Can you see the doodah in the middle of the elephant? Yeah, stare at the centre of that. Don't look left, right, up or down. Just keep staring at that. Okay, and in a moment I'm going to remove the image, you're going to blink a couple of times, and then you're going to make an R noise. Are you ready? Blink a couple of times. I can hear the R noise. That's good, that's perfectly normal. Different parts of your retina were getting bored at seeing those colours, and so they switched off. They literally became blind to those colours. And so when you see nothing, you see the opposite of that colour. Should we do it again? I've got a different image. Stare again at the centre. Keep staring. Don't look left, right, or up or down. We don't want that image to be blurred. I want to burn this image into your retina in a non-blurred fashion. I guess you recognise the shape. Don't look at the shape, just stare at the thing. And I guess you can see where this is going. Okay, blink a couple of times. It's wherever you look. It's on the ceiling. It's on your neighbour's face. It should go after a day or two. Can you tell me what the blue turned into? Yep. Can you tell me what the yellow turned into? Blue? Yep. Can you tell me what the black turned into? Yeah. Every colour has its opponent colour, an opposite colour, the evil other half. And designers know about this colour, and they go, you can't get any different between this colour and its complementary, its opposite colour. The scientist Maxwell used these after-image colours to analyse how people saw colours, and people with different vision defects, colour blindness, see different after-images, and from that you can work out how we see colour. I did this talk in Belgium. I felt I should use the Belgium flag. It didn't work so well for me. The audience were all going, ah, but I just didn't. It took a long time for me to see the colours. And that got me thinking, it's not just what you're seeing, it's what you think you're seeing. It reinforces what you see. And a little bit later, after doing that talk, I was walking down the road and I saw someone with a leather jacket on with the Union flag on the back. It was late-ish at night, and from a distance with the ill illumination, I wasn't quite sure what I was seeing, but I could pretty much see the colours. But when I got really, really close, I found out that there were no colours on there at all. It was just in black and white, and I was imagining those colours. You can see things that are not there. Do you remember that first slide? We're working slowly towards that. Colour's quite complicated. If we look at the response of the human eye, there it is, and it looks like someone's thrown a psychedelic bedsheet over some furniture. And they have. There are some details missing. Let's remove that and look at the details. We have 3 types of sensor in the eye, responsive to different parts of the spectrum by different amounts. There's one line there that shows that something is responsive more to the blue end of the spectrum than the others. There's another one that's more to the red end than the others, and there's something more responsive to the middle part of the spectrum. And these 3 different signals from this mosaic of sensors in the retina gives us the sensations of colour. Colour is a 3-dimensional perception. And this is a machine that you can use to investigate how we perceive colour. This was built by my boss, Julie, in the 1980s, and there she is doing some experiments. She was repeating experiments that were done in the 1920s by WDW, as he was affectionately known, and John Guild. And this is what they did. To see light, to see colour, you need light in the first place, and that accommodation level will affect how you perceive light. So we set a sort of general field at daylight viewing conditions. We took a spectrum, and we grabbed just part of that spectrum. In this experiment, we're taking the cyan part of the spectrum. And then we ask observers to tell us how much of 3 primary colours, selected from different parts of the spectrum, are needed to match that colour. Can you imagine in your mind how much you'd need of blue, green, and an orangey red to make cyan? Let's have a go at doing that. If we had equal amounts of the first 2 primaries and no red, we'd actually get a very poor match. So observers in the 1920s and in the 80s were allowed to twiddle the knobs and adjust it to create a decent match for them. And it doesn't always work first time, but eventually you'll get a reasonable match. Now, can I tell you that the actual settings on those dials will vary by person, by illumination level, by eye. But you can end up getting an appreciation of how individuals see or sense colour. And in the 1920s, those data were combined to create what we now call the standard observer. That's the data all pooled together. And we created a colour chart based on the human perception of colour. There it is. And in 1931, an international committee agreed that this is the chart that we will use from now on. And today, that human model, based on those 17 observers, is embedded in cameras and projectors and television and iPads and any colour rendering system. Even traffic lights. Oh, I did say that colour is a 3-dimensional thing, but this is a 2-dimensional plot. We're losing something, aren't we? There aren't 3 numbers being represented here. What we are not representing is the lightness of the thing. So this is sort of a slice through a piece of psychedelic ham with lightness coming out of the paper. You can imagine the 3rd number telling you something about how bright it is. Anyway, traffic lights. There's a British standard about what the colour of traffic lights should be. There they are. The reds should be in that little red area down the bottom, the greens there, and the ambers there. Why? because we don't want people to confuse the different colors. Now, you can actually plot color blindness, color confusion lines for people with color defective vision on this chart. Let's pop them up there right now. There they are. And can you see how everything along each one of those black lines, that would be confused as being a similar hue for someone suffering from deuteranopia, a type of color blindness. Can you see that there's no line connecting the red with the green? Can you see that? So that's great. Those people suffering from that kind of color defective vision won't confuse the two types of traffic light. But there are three types of color blindness because there are three types of sensor in the eye, and there are three things to go wrong, and those are the other charts as well. How do we measure color? Well, we could take a photograph of the spectrum. Here we have some white light coming in and shining onto one of these gratings, like I just showed you here, and you can take a photograph of what comes off. And you can compare the photograph of the spectrum with a photograph of a standard spectrum of a standard lamp that you know how much light is coming out at each wavelength. And from that, you can infer exactly how much light is present and then pop it through a model of the human eye, and then you can plot it on that color chart. Alternatively, you could do the cheap and easier version, which is just measure the amount of green light, the amount of red light, and the amount of blue light, the same way that the eye does, get out three numbers, and again, that would give you a reasonable approximation. This is the way that cameras work, of the color that you can see. And so we can use that to measure creme brulee, or whatever you like. But perceived color is oh so much more than just amounts of red, green, and blue. This is the second part of my talk, and this is why I basically stopped measuring color, because it just becomes so complicated. It moves from physics to biology and psychology. And so let's do another experiment on you. By the power of PowerPoint, I'm going to rotate a piece of white so that it obscures these little dots one at a time. It's going to do that, okay? Can you see? So let's do it. And while it's doing that, I'd like you to stare at the cross in the center, and something weird will happen. Keep staring. Keep staring. It'll vary. The experience will different. People will be different. But what a lot of people see is that initially you see a kind of green thing going around, and then maybe you only see the green thing going around. Too weird. I'm not going to explain that one. It's too complicated. I'm going to tell you where all these changes occur from, though. So we have a mosaic of red, green, and blue sensitive cells at the back of the retina. And exactly how they're distributed and the proportions of them will vary, and that's controlled by your genetics. Someone took a photograph of the different types of cell in the back of the eye and colored them in with a felt tip pen. That they really aren't this color. This is just to show you the different responsivities of different parts of the eye and different people's eyes. And so this really does mean that different people can sense color differently. I built a machine to measure how shiny cats are, because someone wanted me to. And when I did that, I realized that we don't see light linearly. Different equal steps of increase in light don't appear to us as equal increases in light as perceived. We have a very non-linear response to light. So there I've got the whitest thing that the projection system will display and the darkest thing that it will display, labeled 100 and 0. Where do you think 50 will be? Let's put it in. Does that appear to be exactly halfway between the other two? No. Again, we can do experiments and you can start messing around with it, but we have a non-linear response to light because we have to accommodate such an enormous range of light levels. Age. There's my dad and my daughter, many years between. Now, my dad's eyes have been radiated by ultraviolet radiation for a very, very long time. So they've yellowed with age, which basically means he's not getting so much blue light going to his retina. So for him, if I divide the picture on the left-hand side, he'll see everything literally through sepia tones, whereas my daughter will see blue things a lot brighter. Light level. We have another type of sensor in the eye responsive to low light levels, which actually gets more excited by the blue end of the spectrum. So if you go into the bluebell wood late at dusk or at dawn, there's a reason to do it, you'll see that bluebells look suddenly a lot brighter, and yet they look less colourful. So the way we see colour varies with light level a lot. Have you ever done decorating? Did the colour come out the way you wanted it to? There's two things going on at least. One is that as the light comes in, it gets mixed around, and so it will affect the other colours in the room. The other thing is that when you're looking at a colour swatch, you're seeing an area that's different to the whole room being painted that colour. The majority of the colour sensors in the eye are located in the centre 2-degree field of vision. That's a thumb at arm's length. That's where you're most sensitive to colour. Beyond that, there's not so much sensitivity. So depending on the area of the thing you're looking at, you will literally respond to the colour very differently. So I did a kind of mind experiment. What do we actually see? What's actually going into the brain? Well, the first thing is that the eye is a pinhole camera, so the thing's upside down. Secondly, if we mark in 10-degree slots, there's a blind spot. I've marked that in black in this image, where most of the light, well, it's not being received at all because you just don't see it. The cabling carrying the signals to the brain obscure it. And then also, can you see there's a little white disc somewhere in that picture? That's that 2-degree field of view. That's the bit that is most colour sensitive. So really, that's what's going into the brain. What an amazing fix-up job that brain is doing for us. It remembers what's going on. For fun, we used to exploit this chromatic adaptation property. At MPL, where I work, we had a big machine for measuring light bulbs, a big sphere that you could walk inside. And for fun, we used to put a green light bulb in there. People would go in, they'd be amazed at the fact there were no ceiling, floor, or wall, and the acoustics were great. And while they were in there enjoying it all, their eyes were getting bored with the colour green, so when they came out, everything looked pink. Colour constancy is an odd one. When you look at a scene, you refer everything to the whitest thing in the image. So here, I guess it's some of the lettering. And things look greener or yellower or bluer than that. And the eye is very good at adjusting. So again, the banana in this scene looks the yellowest thing and the orange looks orange. But in an absolute sense, in this image, the grape and the orange are actually identical in colour. And the two colour blocks down the bottom are actually identical in colour. So the eye does some pretty odd things, or the brain does some pretty odd things. And in fact, we have things called special colour cues. So bananas always look yellow, independent of what colour they actually are. Some great titles of scientific papers. If it's a banana, it must be yellow. The role of memory colours in colour constancy. Nearly finished. I've got a few more little curious demos to show you. This is such a good one. It got nicked. I had to take a photograph of it. Do those two bits of pink look the same to you? They kind of do, kind of don't. The more you stare at it, maybe they do, which is interesting. It's all dynamic. It varies with time. Because of what they're next to, colours affect each other. And in fact, those pinks are identical, but... and then if you stare at it for long enough, they appear to be the same, and then if you look away and look back again, the image comes back. The effect comes back. And it varies with field size. So if I do that, they really do look very different indeed now, don't they? Artists know about this and they exploit it. So Van Gogh used to have green next to orange because they made the orange look more orange and the green look more green. And yellow stars in blue skies, so that the yellow looked yellower and the blue looked bluer. This is a really weird one. The areas labelled A and B, do they look the same? No, they are. If I put that there, I've got a block there, and the area labelled A right next to it looks the same, and B looks right next to it, and the thing is uniform in itself. So they really are identical in colour. Would you ever, would you, it's crazy, isn't it? Three things going on here. B is in shadow. Things in shadow look lighter than they really are. We open up the lightness of things in shadows. B is surrounded by dark things, so it looks lighter than it really is. And the third one, this is the weirdest, this is the one I worry about most. You recognize the pattern. You know that B is light and A is dark, and what you know is actually influencing what you're seeing. Is that mind control? Is that scary? If you wanted to get machines to see color and light the way that we do, you'd have to program all of this into them. You'd have to measure the contributions and the effects. You'd have to get the machines to recognize the patterns and measure things that are around the things that you're looking at. This is my last slide. It's quite a complicated slide. I'm going to move that there. Can you see that the green next to the green is green? But if I shrink it down, we have a... Does it still look green? Depends where you're sitting, depends how old you are, depends on so many things. What about this one? It is the same green. The only thing I'm doing is changing its size. But does it still look green? Does it look the same green? No. So depending on where you're sitting, I mean, that might look a bit like that. That might look a bit like that. Again, we can tweak this for personal taste. It gets worse. Look at the eyes. Do the eyes look the same? If I do this, maybe I'll say those two are the same, yeah. Maybe the eyes are a little bit darker, but does it look cyanish in hue? Or does it look grey? You can see where this is going. Those two eyes are identical. So, we see these identical. This is where I began. This is where I'm going to end. Thank you.